other point I wanted to make is I wanted to talk about CAH. It's congenital adrenal hyperplasia. This is the most common cause of ambiguous genitalia. And this, like, you're missing at one, and there's a few different mutations that it could be, but you're missing an enzyme in like creating steroid hormones. So you end up having deficiencies in different steroid hormones, which includes your sex steroids, but also includes your like cortisol and fludrocortisone, which is like a mineral corticoid, so it regulates salt in your body. So babies with this can have what we call salt wasting, so they have really, really low sodium, which leads to low water as well and dehydration. They can also have like issues relating to the less cortisone as well. Like they just won't be feeding, they'll be sleeping, they could die from this. So this is like really, really serious. So when kids are, if a kid presents with like ambiguous genitalia, they're always tested for this first, just to make sure that they don't have these other hormonal issues as well, because it's treatable. You basically just have to give them pills for the rest of their life. And like the pills are kind of, because I'm in pharmacy school, I have to talk about them a little, but like they're kind of like a bit annoying because you have to change the amount of dosing depending on activity. So your fludrocortisone, which is related to like holding on to salt and water in your body, if it's like 40 above and you're working outside, you have to take a lot more because you'll have too much salt loss otherwise. And
is the CAH then a gene or is it an enzyme that triggers?
CAH is the name of the condition. There's a few different mutations, and there's like, they all have silly names. It's like, give me a sec.
So it's not like the name of a process or of a string or anything, right?
It's because it's like the metabolism to make these hormones. There's like different enzyme pathways, so it's like problems with those enzymes. So you have, I think one's like 21 hydroxylase or something is I think that's the most common one. And then there's just other ones in the process, but depending on like which enzyme in the process and like how bad the deficiency is, like do you have like one copy that's just completely useless? Do you have one copy that like works a little bit? And yeah, so like how, and the other issue is that it is X-linked. So that means it's on your X chromosome. One of them, at least one of the genes is on the X chromosome. There are other ones that are not on the X chromosome. So again, it's like a family of genetic diseases that present similarly. This happens relatively frequently where like the disease looks the same. So you classify it all in the same group, and then you do more research and find out it's actually like several different genetic anomalies that's causing it. But
so in talking about all these interactions, sorry if I keep stopping you, but I'm just trying to get a clear mental picture of it. From my understanding, most of these molecules interact with lock and key mechanisms, like where one has an opening that only fits a certain other opening, and that's how the two enzymes or whatever bind one set of solutions to another set of solutions. Is that always the case? Is it always like a problem with the lock and key combination? Or
in this case, well, it's not always, because you can get like things that so basically these enzymes that are involved in the process. So you have your like molecule that you're trying to make in a steroid molecule. It's got a 4-ring structure. It's very distinctive, but it's like all the stuff on the outside of that 4-ring structure is different for each one. But basically that fits perfectly into an enzyme, like active site is what it's called. And then the active site makes the molecule sit in such a way that the reaction that you want to happen happens exactly the way you want it to. So then that like allows it to move on to the next step. But
so it's almost like its physical form is determining the actual response action.
That's exactly what it is. Structure determines function with proteins. That's like drilled into our mind. That like if you get like one change, like one different amino acid, so I didn't really talk about amino acids, but proteins are made of amino acids, and like the DNA like codes for like, here I want an alanine, then I want like whatever other, I can't think of it, valine here, and then whatever. You just keep adding more amino acids into a chain, and then the chain has to fold into a very specific way, which is a complex process. Very complex process that
we'll be talking about that in an upcoming episode of Neural Nets, actually.
Oh, exciting.
Foreshadowing.
Okay. Ooh, yeah, they had that new AI come along. That's really, really
AlphaFold.
sweet. Yeah, so anyway, so you can eat, so if you have like something wrong in the active site, then the enzyme is just not going to work at all. So let's say you have the wrong amino acid like on the other side, and it was like supposed to be a basic acid and now it's an acidic acid. Like that's a problem. But I know saying basic acid doesn't make sense, but like they're called
simple acid.
They're still called amino acids, but like they use an acid-base reaction to like hold themselves together, and then like they have another part coming out of it that determines the different structures, and sometimes they have like an acidic group and sometimes they have a basic group, and that also determines the structure of the protein and like the function as well. But like if you've got like a really wrong thing on the other side, it might affect the folding of the protein, so maybe it doesn't fold quite right, but it's still close enough that it can kind of do its job, but poorly.
It's just kind of gimpy.
Yeah, it's just gimpy. And there's other ones that you have that you can like get a stop codon is what it's called. So basically you might have like, I don't know, like 80 amino acids in this gene, but the third one is just actually a stop now. So you just create this chain of 2 that just gets eaten up. So that's like, it's the stopping too quickly. So that's completely not functional.
Is that similar to acting like a telomere?
Not really. Telomeres are like big, long things that are on the end of your chromosomes that just prevent it from getting eaten and falling apart. So
those are more like knots or braids?
We're talking like telomeres are like on the structure of chromosomes, and right now we're talking on the structure of individual genes.
So tiny fractions of chromosomes. Then.
You do actually have, because now we're getting more into virology here, but like your body doesn't like random RNA floating around because viruses often use RNA. So it'll chew it up. So basically any RNA that's coding or like useful and necessary in your body is like specific. So like your messenger RNA, which will code for the protein, will have like a bunch of A's on the end and will have a cap on one side so that your body doesn't chew it up right away. It's a viral defense mechanism.
But that's different from a stop in what way? Like, can you compare those two?
Because a stop just basically tells the machinery, the translation machinery, which is your ribosome. And your ribosome basically it reads the mRNA and then puts the protein together. And the stop codon just tells the ribosome to go away. So your mRNA is still there, but it's just, instead of saying put an alanine here, it says, okay, my protein's done now. So then the machinery just leaves, and now your protein is 2 amino acids long and is just going to get chewed up.
The rest is scrapped, or is it reused?
It's reused. Everything's reused.
Oh, cool.
Until it can't be. And then it's still. And then you just poop it out, I guess.
Do we poop out our bad DNA? Like, sounds like a stupid question, but...
DNA has a lot of bacteria in it, and because bacteria cells are small, they have a lot of DNA. So you do poop out a lot of DNA. And a lot of
just a really cool fact. If nothing else I take away from today,
you also can't eat bacteria because there's so much DNA. It's too nitrogen rich, which is why bacteria is not a good source of food.
Which is unfortunate, because it would be a great source of food if it could be.
Yeah, we can't handle all the nitrogen in it. There's just too much. So